It's my great pleasure today to talk with Jimmy Moore. He's a mechanical engineer. So I'm going to read his background. He received a Bachelor of Mechanical Engineering 1987 and his Ph.D. in 1991 from the Georgia Institute of Technology. He moved to Texas A&M, where he met David Zawieja, and he started to work with the lymphatics. So he was professor of biomedical engineering and director of graduate studies in Texas A&M. And then in 2013, he joined the Imperial College in London and became director of research for the Department of Bioengineering. He started with cardiovascular, working with stents and implantable device, atherosclerosis. And since probably 2004, he worked with the lymphatic system. And that's what we're going to talk about: pumping mechanism, immunity, lymph nodes, and also some of the device he worked on and solution, bioengineering solution for lymphedema. Was this correct, Jimmy?
That's very good, and very kind of you to give me that introduction. Thank you.
Thank you. So I thought we could start with anatomy, physiology, some of the work you did in A&M and in London with your lab. Do you work exclusively on the lymphatics now?
Yeah, it's pretty much exclusively lymphatics. I still have one cardiovascular blood flow project going on in the lab. But yeah, it's I'd say 80% of what I do research-wise is in the lymphatic system.
Great. So I know you have some slides on the anatomy of the lymphatic system.
Right. So here's a diagram I use to explain the basics of the lymphatic system. As you know, Bruno, it's not a common topic. Generally, if I give just a scientific seminar somewhere, I have to explain what it is first. So you can see here it sort of runs in parallel to the venous system, returning fluid. And you mentioned I have a mechanical engineering background, and this slide is very much from sort of that mechanics point of view. So if you think about, the amount of cardiac output, it's 5 liters per minute at rest in an adult human. So that's more than a gallon per minute of blood, which is quite viscous fluid. Pretty much everyone knows their blood pressure is between 80 and 120, and the units are millimeters of mercury. Your aorta is about that big around. It's a pretty big tube. And Reynolds numbers, it's a dimensionless quantity for fluid mechanicians that describes the likelihood of turbulence. So Reynolds number about 2,000, 3,000 and above would indicate that the flow is likely to be turbulent. Most blood flow is not turbulent, thankfully. Turbulence is really complex, and I'm glad I don't have to deal with it. In comparison, you got about 5 liters per day instead of 5 liters a minute of lymph flow. Pressures in the lymphatic system can actually range from very heavily subatmospheric to just barely above atmospheric pressure, which is a really interesting aspect of the physical operation of the system. You can see the largest lymphatic vessel is thoracic duct, about an order of magnitude smaller than the aortic diameter, and the Reynolds number is under 10. So this is highly viscous flow. So turbulence would never really occur there. So when I look at the lymphatic system, I find it to be this fascinating way of moving fluid around the body. And as it does that, of course, it plays an important role in immune function, spread of cancer, and really something like, 8 of the top 10 causes of death in most Western countries can be tied in some way to the lymphatic system. So it's really important as well.
Jimmy, tell me more about the Reynolds number. I remember it's like the viscous and inertial force in a liquid, something like this.
Very good. Yeah, that's right. It's the ratio of inertial to viscous forces. So if the viscosity of the fluid is dominating, so if it's a very viscous fluid like maple syrup, it has to be moving really fast to generate turbulence. So right. The Reynolds number is going to be smaller for, say, maple syrup than it is for water or air.
Sometimes the pressure can be higher than 10 millimeters of mercury in the thoracic duct. It's very funny because some research make it high, some research make it low.
Yeah, it certainly would be transiently because the return point in the subclavian vein is at about 10 millimeters of mercury pressure. So occasionally has to go higher than that to get fluid into the blood system.
Exactly. Perfect.
Yeah. But this slide gives you some idea of the range of research going on in my lab there. And, we've done a lot of pumping analysis in collaboration with Dave Zawieja, as you mentioned earlier. We also look at how the cells in the lymphatic vessel walls sense their mechanical environment and then respond to that. Say, if the flow is not moving very well, it senses that and then triggers more pumping to get the fluid moving again. And each individual lymphatic vessel does that, senses its own mechanical environment and then changes its behaviors in response. So each vessel has its sort of internal feedback control mechanism. And then we're looking at mass transport. So transport of nitric oxide in lymphatic vessels is something that we've done. We've also looked at transport of chemokines. So these are cytokines or substances that make cells move. And the ones we're interested in are targeted towards immune cells. So when an immune cell captures some antigen, then it needs to find its way into the nearest lymphatic vessel and migrate through that system and get pumped along with the flow. And that movement of that cell is particularly in the periphery, is mostly guided by chemokines. So we've been looking at mass transport of chemokines and how that influences cell migration. And then the movement of fluid and chemokines through lymph nodes has been of interest to us as well. And in terms of practical applications, of course, understanding the basic operations system is a lofty goal because it's a very complex system. But we want to apply that in some way, and vaccine design is one of the ways that we're looking to apply our research. So a vaccine often needs to include something that kicks up your immune response called an adjuvant, and then that affects lymphatic pumping. So with that, and again, let's work with Dave Zawieja. We'd like to know more about the way those things work.
So let's go back to mechanotransduction. Are you talking about, for example, the endothelial cells that could be contracted, pressed from outside, and that will create the pumping, or even from inside, fluid enter the lymphatics? That kind of mechanotransduction?
Yeah, so the endothelial cells would be one of the two cell types that are in the lymphatic vessel wall. So lymphatic muscle cells occupy kind of the outer layers of the lymphatic vessel wall. The endothelial cells would be on the inner layer, and they would be the ones that would be sensing the flow. And what we think they do is they sense the frictional force of the fluid flowing past the wall, and they transduce that in some way. Transduce that mechanical signal, which is where the term mechanotransduction comes from. And that changes their geometry, that also changes their protein expression, and they can secrete substances that either contract the lymphatic endothelium itself, or they can change the contractile behavior of the lymphatic muscles that actually produce the contractile pumping action of the lymphatic system.
So we're talking mechanotransduction. Did you find any receptors inside of those muscles?
So the lymphatic endothelial cells, there's been a lot more work done on mechanotransduction of lymphatic endothelial cells than lymphatic muscle cells.
Which one feel the compression from outside? It's the receptor on the muscles or the endothelial cells?
I suspect it's both.
Okay.
So blood endothelial cells, it's been shown that they can respond to fluid shear stress, as I mentioned earlier, and stretching. And we've done that research going back into the 1990s, and other people have looked at blood endothelial cells as well. Lymphatic muscle cells, lymphatic endothelial cells, I suspect they can also sense flow and stretch, although no one's really gotten that far with those cell types. The lymphatic muscle cells would not be directly exposed to the flow. So I think if they're sensing anything, it's going to be the level of stretch, and that would certainly depend on any compression from external tissues movement.
Oh, muscle contraction, skeletal muscle, smooth muscle, beating of the artery, breathing through the diaphragm, all those external compression may influence more the muscles of the lymphatic, in the lymphangium.
Yeah, they provide, in some sense, some pre-contractile motion to drive lymphatic flow. But yeah, the lymphatic muscle cells in the walls of the lymphatic vessels, some people call them smooth muscle. But they're actually not pure smooth muscle. They're kind of a mixture. They've got some striated muscle component into them as well.
They say they have a mix of the three type of muscles. It's a fourth.
Yeah, cardiac as well. Yeah.
But maybe we'll talk about that a bit later. So when we have mechanotransduction, but we could not isolate any specific receptors. It's just something we see.
Yeah, I think it's probably multiple receptors. I've always thought that mechanotransduction in any cell is probably relies on redundant systems, just from a safety point of view, if you think of it that way. Some of the G protein coupled receptors are known to be mechanotransductive. There are stretch sensitive ion channels, for example, that might respond to either stretch or flow, shear stress. And then there are primary cilia in these cells as well that likely respond to flow. So we're not sure which of those are active in lymphatic endothelial cells, but I suspect more than one of them.
Right. So maybe we can see the next slide, and then we'll talk about the type of muscles. We are talking about how they combine the three types of muscle in the body.
Sure. So this is really how I got into lymphatics. Dave Zawieja came to borrow a high-speed camera so they could take these movies. I'll show you the movie. So you can see here the lymphatic vessel in the middle. There's a blood vessel below here. It's a vein. And you can see the immune cells going through the lymphatics vessel here. You can see they move forward a bit. They sit still. They move backwards by a slight amount. And the vessel contracts. So you can see it's a very dynamic flow environment. We're actually, what we got involved with here is actually tracking the motion of those cells in there to estimate the fluid flow velocities. And it taught us a lot about the fluid mechanics inside the lymphatic system. We were able to estimate the flow rate. Down here, you can see it's 200 microliters per hour. So if you think about how, a liter is about like this, right? And a microliter is 1/1,000,000th of that. Right. So it's a tiny volume of fluid being moved per hour through this single lymphatic vessel, which is about 100 microns in diameter, which is about the diameter of a human hair. So this exquisitely small vessels do this just fantastic job of pumping fluid around the body. It's great.
But Jimmy, when we look at it, you assume, I know there's all kind of different contraction. You're going to probably talk about different type of contraction and where and how. When you see the compression, you assume that it's going to squeeze it and move forward even faster. What you see here is the contraction almost stop the flow, make it go backward.
Yeah, it's important to keep in mind, not only do you have a valve just upstream and just downstream of the frame of this movie, but the vessels upstream and downstream are also contracting, probably not in sync with the contraction of this vessel that we can see. So there's a lot going on here. So you've got, when the one upstream contracts, that'll tend to push fluid through this one. When the one downstream contracts, that tends to send fluid a bit backwards. So,
yeah, in other vessels, you see that during contraction, the flow is propelled forward. This is just
and the valves are super interesting as well. So I'll start this other movie over here. This is a three-dimensional microscopic view of a lymphatic valve. So they're bileaflet valves, meaning they have two leaflets. And you can get an idea of how they prevent flow from going backwards. So this is in the open configuration. Flow would be coming at us in this instant of this view here. And then if flow tries to go back the other way, those two leaflets would slam shut like that to prevent that backflow.
So why are they interesting? I mean, they call them bicuspid, just for the people.
Yeah.
So they go, why is it more interesting that, the heart valve, they have three of them, sometimes two of them? What's so specific about those valves?
So they operate, you remember the Reynolds number we were talking about earlier. So the aortic valve, for example, there's a lot of not exactly turbulence, but sort of spiraling flow around the aortic valve that's very important to its operation. At these lower Reynolds numbers, you don't get all that spiraling flow. And so these valves have to operate in a very different fluid mechanical environment from the heart valves. It does require, some pressure difference and a bit of inertia to close these valves. But yeah, they're also very thin walled compared to heart valves. So it's basically just a single sheet of elastin and a few cells on both sides. And there's a bit of bias in the open direction, which is something we've published several scientific papers about. If there's no pressure difference across the valve, it'll just stay open, sort of barely open configuration, much like the microscopic image you see here. But as soon as flow starts to move back the other way, that it does shut pretty quickly.
So this is in a rat, right?
That's right. So this would be in the gut of the rat, in the mesentery of the rat.
Mesenteric vessel in a rat, yeah.
We know that there are electrical connections not only within the lymphangion that we can see contracting here, but also from one lymphangion to the next. There's some electrical connection, but it's not always triggering coordinated contractions between the two adjacent lymphangions. So there's still a lot to learn about that system and how it sort of controls lymphatic pumping.
In a vessel that's pretty long, it doesn't go in a coordinated manner? It just all the lymph.
Sometimes you would see it going, and if you've got say 10 lymphangions in a 1 centimeter long vessel, you might see some coordinated action of it going downstream or going upstream, or maybe one of the vessels in the middle is relaxing and not doing any pumping at all. It's not a carefully coordinated system, although it can have coordination to it. It's not a very efficient pumping system. Yeah. So every time we try to look for, and this is part of what we do with the mathematical modeling, every time we try and say, all right, well, we have a hypothesis that there's some optimum efficiency that the system achieves by the way it coordinates its contraction, it doesn't really work out that way. I think the system is not really designed for optimal pumping. It's designed for keeping generally things moving in the right direction.
Did you try it with under high volume flow to see if they are more coordinated, more efficient if you have a higher volume?
Yeah, we've done that. So we had a Ph.D. student provided an extra fluid load to a certain area of the mesentery, and we looked at how it changed the pumping. So yeah, it increased the frequency of the pumping. And that's the mechanotransduction in action there. But really, it didn't get any more efficient.
Wow.
It just was pumping faster.
That's why we assume that those lymphangion activity, the lymphangiomotoricity, needs some help, specifically with older age in human. You need the muscle contraction around it. You need to move. That's why it's not so efficient by itself. You need the rest of the indirect way to make this lymph move.
Absolutely. And I think most of the lymphatics in the body get some benefit of movement of adjacent tissue to keep the flow moving. So I think you're right. The reason the system doesn't need to be so efficient is that it gets all this external help essentially for free.
That's why we get wheelchair edema. People that don't.
Keep moving. Good advice. Lastly, here I'll just point out that the thing at the bottom that says 8 orders of magnitude faster than cell crawling. So these velocities of the cells moving through due to the fluid flow in this lymphatic vessel are about 1 centimeter per second. If those immune cells had to crawl themselves through interstitium or along the vessel of the flow, in other words, if we cut off the lymph flow, it would take them 8 orders of magnitude longer time to reach, say, the lymph node downstream. And 8 orders of magnitude is basically the analogy I give is if you take the velocity, you can walk. So you can walk about 3 or 4 miles per hour, right? If you go up 8 orders of magnitude from walking speed, you get to the speed of light.
What? Wow.
So that's how much faster it is to move cells in the lymphatic system than it is to depend on them crawling to get where they're going.
Wow. Did you see with Agachev in Texas the difference with human?
Yeah, in a similar sized lymphatic vessel in a human, I think these velocities would be very similar. So this is a simple animation that I made to describe how the lymphatic system pumps. So I'll just run my little animation here. So you've got the vessels contracting, you've got the valves, the little things in blue there. You can see them opening and closing as the vessels contract and relax. And basically, the lymphatic system does what all pumps do. It takes fluid from a low position and moves it to a higher position. So if you didn't have the pumping, flow would go from the high to the low pressure. So with the input of energy, the lymphatic system of the pumping energy, the lymphatic system creates potential energy by moving fluid from a low position to a high position. And that's the animation that you see here. We can do the experiment of taking a lymphatic vessel out of the rat mesentery and cannulating it. In other words, sticking tiny, tiny little tubes inside the vessel. And then at that point, we can control the pressures upstream and downstream of the vessel to see how it changes its behavior. That experiment's what's shown in this slide. So this actually comes from Mike Davis's lab. So he used to be at Texas A&M as well. Now he's at University of Missouri. And you can see the valves in this case. So there's an upstream valve, there's a downstream valve. So what you see when there's no pressure difference across the valves, they just kind of open and close, not quite randomly, but they're not very well coordinated. Once we give this vessel a challenge by giving it a higher downstream pressure, you can see that they operate out of phase with each other. And this would be like the heart with the mitral valve opening and closing opposite in synchrony with the aortic valve. So, and that's what you want in this kind of pump is when you squeeze, you want the upstream valve to close and you want the downstream valve to open so that when it squeezes, all the flow goes downstream. So that's an evidence of the mechanotransduction that the vessel is sensing this higher pressure and is wanting to pump more frequently. So you can already see the frequency of contraction is going up for this vessel.
Yeah, perfect. So talk about these different, how specific those muscles are, a mix of skeletal, smooth, and cardiac muscles.
So smooth muscle, like you have in arteries, contracts, on the order of like tens of seconds. So it's very slowly contracting muscle type or so.
To contract and then 10 more seconds to relax, something like that?
And it typically wouldn't contract the vessel shut. It would basically just be a way of modulating the diameter in small amplitudes to control the flow downstream.
Basically. Yeah, they would contract basically when you cut them. They would contract a little bit, but they are not doing regular like that.
Yeah, so the large amplitude fast contraction, and this movie is in real time here. So the large amplitude fast contraction that you're seeing here is due to the striated or skeletal muscle component inside the lymphatic muscle cells. And the cardiac muscle would behave in a similar way, a similarly fast contraction.
That's basically a fourth type of muscle. Hopefully one day we'll have that in the books, right?
Well, that's why I'm in the habit of calling it lymphatic muscle, because I think it should be classed as a different type of muscle. So,
right, it's a different class.
This experiment tells us flow but not pressure. This experiment tells us pressure but not flow.
So, Jimmy, you found that you cannot measure flow pressure in lymphatic vessels, basically, right?
This experiment, if we try and poke in with a servo pressure transducer and measure pressure in here, it disrupts lymphatic function. And you couldn't do that in general. This is a rat with its mesentery exposed on a microscope stage. You couldn't do that to a human, right? So there are some ways so Tim Federa at Mass General and his colleagues have created a way of imaging very superficial lymphatic vessels and looking at the flow inside them. And that's the only technique I'm aware of that allows you to measure flow in any human lymphatic vessel. But again, you wouldn't be able to poke in and measure pressure at the same time there without disrupting the flow. And in this case, we can determine what the pressures are, but there are no cells flowing through here for us to track the velocity. And we tried putting, little fluorescent microspheres in here. We tried putting cells in there, but the cells either float or they sink or they clog the pipettes, and it's just a nightmare to try and measure flow in this experiment. But we know exactly what the pressures are. You have to sort of take what experimental information you have when you're going to build a model, and then you use the mathematics to put it all together.
Amazing. So this flow is going to arrive at the node that's going to create some resistance. And I know you calculate those resistance.
Yeah. It's a very interesting design feature of the system. So the rat mesenteric vessels are all prenodals. Their job is to deliver fluid and cells to the lymph node. And you see this all the time with lymph nodes, that when those prenodal vessels arrive at the lymph node, they start to split into multiple parallel pathways. So you can see the vessel on top splits into three here. The one on the bottom splits into three different vessels there. As an engineer, I look at that and I can see exactly why it's being done. The reason for that is that these vessels that are doing the pumping, so keep in mind they're about the diameter of a human hair. They're quite small. They can only generate a few centimeters of water of pressure, and they have to drive flow all the way through this lymph node here. The lymph node is packed with millions of immune cells and a very tight collagen matrix. It's like trying to pump fluid through a very tight sponge with very tiny holes in it. So it takes a lot of pressure to do that. And these things just can't generate that much pressure. So what they do is they split into multiple parallel pathways to spread the flow out into multiple parallel pathways so that it can still get the flow rate through there at a lower pressure. Proper way to express this is that if you just had a single vessel, it would have a resistance of, say, 6 units. If you have 3 parallel vessels here, the resistance presented to the system is 6 divided by 3, 2 units. So you can cut down the resistance to flow by having multiple parallel pathways for flow going through there. And that's exactly what these lymphatic vessels do as they approach a lymph node. the efferent vessel, the outgoing vessel here, can generate its own pumping. So if the pressure is zero at the beginning of the efferent vessel, it doesn't really care. It still keeps fluid moving in the right direction. Dave Zawieja keeps telling me he's seen evidence that lymph nodes can contract. I've never seen it myself, but let's just say that it's possible. But still, to drive flow all the way through this thing that's tightly packed with all those cells is a real job. I think it's almost certainly the afferent vessels that are moving all the fluid through the lymph node. But I will allow for the possibility that the lymph node is doing some squeezing too.
So the afferent and the efferent vessel all the way are contracting, right? All the way to the capsule. And do they have more strength in the afferent because they need to fight this resistance in the parenchyma?
I'm not aware of any specific measurements of that. Okay. Yeah, it would make sense, but then there's going to be resistance downstream of the efferent vessel too, so it can't just get away for free. It needs to do its work. This gives you some idea of what the lymph node looks like on the inside. So you've got these, in this case, 3 afferent vessels bringing fluid into the lymph node, and then a single efferent vessel taking fluid away. You see the blood vessels going through here. This would be where you find a lot of T cells here, and then these follicles here would be where you find a lot of B cells, and there's the communication between the T cells and the B cells. And this gives you some idea of how tightly packed that's as a sponge, that lymph node is. So that's a high endothelial venule there, and you can see how tightly packed the rest of it is. This is the subcapsular sinus, which runs around the periphery of the lymph node. And mechanical engineer point of view, this is really to spread the flow around the lymph node. So, it comes in these specific locations here, these 3 locations. But then these B cells here need to know what's going on. These T cells need to know what's going on. So the flow kind of gets distributed around the periphery. And then some of it will actually dive into the middle of the lymph node, but you can see there is this pathway for some of the fluid, at least, to move around the outside. And that really got us interested in terms of figuring out a lymph node's, just say under normal, healthy conditions, how much of the flow is going to the middle versus how much would be going around the outside there. And then you've got also some fluid exchange with the blood vessels. So you've got this quite strange and interesting situation. If you total up the amount of flow coming into the lymph, not all of that lymph is going to come out in the lymphatic vessels. Some of it's going to go into the blood vessels. Or is that fluid is retained inside the lymph node and the lymph node swells. So we all know that lymph nodes swell, and I think the shift in fluid transfer patterns between the lymphatic compartment, the blood compartment, and the volume of the node I think that fluid shift plays an important part, at least in the early phases of lymph node swelling. I mean, they can go up an order of magnitude in volume within a day. It's pretty amazing remodeling of that structure.
So under physiological normal condition, the lymph is more reabsorbed by the venous side. Do we know, did you quantify that? Because I heard different numbers. Do you know how much of the lymph is reabsorbed in the...
Yeah, so we have not quantified that, but there were some fascinating experiments done by Arthur Guyton in the 1980s with dog lymph nodes that showed that if you change hydrostatic pressures or oncotic pressures, that you can have these fluid shifts between the lymph compartment and the blood compartment and lymph node. That's been clearly demonstrated. And that's something that we've tried to reproduce in when we construct mathematical models of the fluid transfer there. And we're able to reproduce those experiments.
But I've heard like 30 to 60% sometimes of this lymph is reabsorbed in the blood. That's why you have a small quantity in the thoracic duct at the end, but there's a lot of lymph produced, much more than what you recover.
That's right. So the 5 liters a day that ends up going up to the subclavian veins, there's probably 3 times that being produced in the periphery, but then being absorbed by the lymph node blood vessels.
So what it seems like is a lot of the lymph is going subcapsular pathways, which means the lymph node is only taking a sample of 2%, 10% of the lymph. You just need to understand what's in the lymph. Like you go in your pool, you take a sample of your pool and say, oh, the pH is low. So it does the same thing, and in order to get the response.
Under normal healthy conditions, exactly.
I don't know if it's the time, but you have the very specific vessel, the high endothelial venules in the lymphatics.
Sure, there are arteries and veins and capillaries running through the lymph node. as shown in this illustration here. And so the high endothelial venules would be the specialized vessels that are just on after the capillaries on the venous side. But yeah, there are arteries as well inside lymph nodes.
So the high endothelial venules, they don't have the flat usual endothelial cell. They have like rounded, plump endothelial cells, and then enable the movement directly from a crossing of cells.
Yeah. There's a lot of cell trafficking across the blood vessels inside lymph nodes, particularly in the high endothelial venules. It's an interesting specialized structure. So I mentioned earlier there'd be millions of immune cells, will be mainly B cells and T cells inside a lymph node, and 90% of them would have gotten there via the high endothelial venules.
You have them everywhere. You have them in the secondary lymphoid organ, the Peyer's patch, and the small intestine, the appendix, but you don't have them in the spleen, right?
I've not done a lot of work on that. So yeah,
yes.
you know better than I do, I think.
So yeah, you're going to explain later how also the subcapsular pathway is avoiding this pressure. You have something about that later.
With our mathematical model, we've represented different parts of the lymph node with fluid flow equations. So you can see the flow chart here, where it comes in and can either go through the meaty part of the lymph node where the blood vessels are, or it can go around directly to the medullary sinus from this green area to this blue area here. And whip that all up in our computational modeling software, then we end up with these findings that most of the flow ends up actually going around the periphery, as you just said. You can see 90% of it, in this case, goes around the periphery and goes out the exit. 10% will come in here, and we show it going out the blood vessels there. So it samples about 10%. It makes sure that nothing bad is going on. If it does detect some antigen there, then these fluid patterns change, and that's what we've been modeling as well.
So you can think it's a filter. So you have a lot of metastatic cells. The lymph node, you could assume, will catch most of them and try to destroy them inside the nodes. It's not the case. You just sample a smaller amount. A lot of the metastatic cells are going to go outside too.
Yeah, hopefully they would be detected.
So rather than be trapped inside and, like a fortress and destroyed, there will be a lot of them going to go outside, and maybe the dendritic cell or something, they're going to run after them, or maybe at the next lymph node to try to stop them. But they are not a filter as much as a testing. Maybe there's a way for them to select those cells and bring them inside the nodes. But if 90% of them are going outside, the humoral response is responsible for catching them, not just the lymph node as a fortress and a jail for those cells. They won't trap many of them, as many as we want.
Yeah, honestly, I wish there were more research done on that process. I think it's fascinating and absolutely crucial. Something like 90% of all cancer deaths happen because cells got by the lymph nodes and went somewhere else and set up a secondary tumor.
Right.
So, there are macrophages that live at the bottom of the subcapsular sinus here. And I think they perform some of the job of identifying those metastatic cells and helping to deal with them. Hopefully, some of the antigens would then be presented to the T cells and B cells within here for those things to be identified later on.
So you have 3 types of dendritic cells, at least. I've heard of fibroreticular cells also. Do you know what they do exactly?
Yeah, they live inside the lymph node. So some of that meaty part of the lymph node, there would be fibroreticular cells in here, particularly in the interfollicular zone here. And they probably serve multiple functions. My understanding of them is, in terms of the way they affect flow and the lymph node structure, is that they provide structural integrity for the lymph node by maintaining something like a smooth muscle contractile latch state. So they're holding tension, but not expending very much metabolic demand in order to do that. And then as the lymph node swells, they then go into a relaxed state. And there's been some fantastic work done here in London looking at the relaxation of the cells inside the lymph node as a role that plays in lymph node swelling. But yeah, to me, the lymph nodes are just absolutely fascinating organs. And much of our research right now is actually going into trying to understand lymph nodes better. And I'm of a certain age. I've reached a point in my career where I've found the topic I want to research for the rest of my career, however long that is, and it's lymph nodes. Because I think we could work on these things for decades and still be finding absolutely fascinating discoveries about the way they behave.
So I saw chemokines. You want to talk about the CCL19 and 21, the chemokine in the node, maybe?
Yeah, so all of this fluid, movement of fluid around the lymph node is also transporting these chemokines, CCL19 and CCL21, as you mentioned, are very important inside lymph nodes because they direct antigen-presenting cells to certain parts of the lymph node where they can present antigen to the other cell types inside the lymph node, and that's hugely important for adaptive immunity. What we found in this 2017 paper you see cited over here is that these flow patterns definitely shape chemokine concentration gradients, which is how these cells detect which part of the lymph node they need to migrate to. So they'll generally migrate in the direction of higher chemokine concentration. So if there's a source of chemokine inside this meaty part of the lymph node here, that direct the cells to migrate inward. What we've actually found in our recent experiments, and we haven't published this yet, is that fluid flow itself can stimulate immune cell migration, just in the absence of chemokines. So that's something we look forward to publishing. I'm not sure anybody knew that before. So that's a nice new finding for us. Right. So fluid flow serves an underappreciated role in lymph nodes. And when we published this flow study here that you see on the slide in 2015, it was really the first ever study of flow in a lymph flow through a lymph node. I'm happy to have the first paper on a particular topic. That's a nice thing for a scientist to have, but I wish there had already been a lot of interest in this because I think there's some fascinating things and very beneficial things we could learn from looking at the mechanics of what's going on inside the lymph node.
So when you said 10% of afferent lymph flows into the blood vessels, it's actually probably more than that, right? Not more than 10%.
That was our finding here, and it agreed roughly with the experiments of Guyton that he did in the dogs. It would depend. We have, 500-600 lymph nodes. It's not exactly 10% in all of those lymph nodes, and it wouldn't be the same for my lymph nodes as yours. So I think it's an indicative number. It certainly does vary, probably from flow going in the opposite direction to 30, 40, 50%. So there'd be a lot of biological variability. So when I first started doing research on the lymphatic system and being fascinated by the fluid mechanics of it, as I mentioned earlier, we're always looking to apply the results of our basic science research to solving problems. And I quickly latched onto lymphedema as a problem that I thought maybe we can help solve because it starts out at least as a fluid balance problem. So your audience will be familiar certainly with the fact that 1 in 7 women get breast cancer and about 20% of them end up with a swollen arm as a result of their treatments. So it certainly depends on what kind of treatments they receive. And we've just submitted a meta-analysis of breast cancer-related lymphedema incidents for publication, and we look forward to having that information out there. It's still about 20% overall. Unfortunately, it's not really come down that much, despite surgical practice changing and everything. It's a lot of people affected with this disease. And this QR code over on the right here, I would encourage anyone who is not aware of the severity of lymphedema, please click on that QR code and go watch the movie. So we have a committee of breast cancer lymphedema patients that advise us on our research. This is that group of patients telling some of the stories of what it's like to live with lymphedema. It's about 3 minutes long. It won't take much of your time, and you'll never forget it. It's really impactful. Wow. So this is a serious problem, and we definitely want to do something to try and solve it. So if you remember, these multiple parallel pathways of the afferent vessels coming in, I took that and I used it as a design principle for an implant that we could put in at the time of breast cancer surgery to replace the lymph node that's being taken out. So this is the device. I've got one here in my office. That's it. Don't know if you can tell. I'll hold it up close, but yeah. So it's quite small. It's got 6 parallel pathways, as you can see here. So multiple parallel pathways to reduce flow resistance. It'll keep fluid moving, hopefully, in the early phases of implantation and serve as the guideway for the regrowth of lymphatic vessels. And then these 2 suture tabs out to the side here will keep it oriented in the right direction. We did some early trials in rats with this implant and found that if the implant rotated out of the flow direction, it didn't work. When it stayed oriented in the flow direction, it was actually successful in reconnecting lymphatic vessels. So you can see the design here. It's made of medical grade silicone. which has been around for decades, well known to be a safe material. We can release drugs from the implant if we need to encourage further the lymphatic vessel regrowth, but we are hoping it'll work without the drug release. So conceptually, this is the way it would happen. So here's the lymph node. The surgeon is resecting the lymph node, taking out, leaves behind the lymphatic vessels. We put in our implant, and any surgeon can put this in. It takes maybe 5-6 minutes to implant it. They just have to tie sutures to the surrounding tissue, and every surgeon can tie sutures. So no new training required, only a few minutes added on to a surgery that's already taking hours.
That's 5 minutes per node, right? Sometimes they take off 20 nodes, 50 nodes, so they're going to have to do.
That's right. Sure. So if the nodes that they have removed are adjacent to each other, we would suggest really only one implant to replace that. But if the nodes are further apart, then it could be done like for like in terms of putting in for every lymph node that was taken out. These are actually quite inexpensive to make. So if you think about the way we would charge for the implant, we would probably just charge per patient and let the surgeon decide how many they think they should put in. That's a common thing that we have to think about when we're designing devices like this, is what's actually happening during the surgery. If you think about this in comparison to other techniques that are, maybe used to treat lymphedema or to try and prevent it. So these slides are more kind of business planning slides here. So this is our competitor analysis here. So we're aiming for Faciliflow to be so safe. Again, it's made of medical grade silicone, that it could be used as prevention. So it would be implanted at the time of breast cancer surgery in a patient who might be prone to developing lymphedema later on, but doesn't have it at the time of surgery. So it's a preventative device. And again, implanted in minutes. The cost of bringing it to market is high. So, we have to pay for clinical trials and all that. It's going to take quite a long time and tens of millions to get it to market. So that's what actually determines the cost of the implant rather than the cost of manufacturing.
How long are you before market? How many years?
If things go well, we could be on the market by the end of this decade, but that depends on some funding coming in. So we're actually struggling with funding right now. So as you'll be aware, Bruno, that the lymphatic system is underappreciated. Lymphedema is underappreciated as a disease. There's not much resource devoted to it. And in the current med tech funding environment, funding for any startup is very difficult to get these days.
You are working also on the adjuvant. As far as I know, I remember, we talked about, was it 1925, something like that? Alexander Glennie. He talked about how those adjuvants needed to be added to the vaccine. But as far as I know, it's pretty random and it just works. We don't know why. But my question about that, after you explained this, we don't know really how they work, those adjuvant in vaccine. Do we know how much side effect they create?
Yeah, well, aluminum salt is probably something that's, fairly harmless in the body in the small amounts it's used in a vaccine. There are other classes of adjuvants that have more serious things in them that can be toxic, right? So there are plenty of things that medics do without knowing why. I think that's underappreciated in medicine. It's called practicing the art of medicine, not executing the exact science of medicine. And there are some things that people do just because they're known for hundreds of years that it works, right? So vaccine adjuvant, as you said, it was accidental discovery about 100 years ago. People know it works, and that's what we do. As an engineer, I want to optimize stuff. So I want to optimize vaccine and make sure that when we do use them, that they work as well as possible.
Do you remember some of those vaccines are given at day one on newborn? Yeah. So you have to work the one immune system reaction, irritating the system to create a reaction. It's good on one side, but how much do you irritate a 1 day old kid?
There's no rational basis for optimizing a vaccine adjuvant. So, let's say you developed your antigen, whatever version of the antigen it is, you need to trigger the immune system to respond to it. That's the job of the adjuvant. But which adjuvant do you choose? Once you developed your antigen, do you pick, aluminum salt or any of the other classes of adjuvants? And even there are subclasses. So which of those do you pick? And there's no rational basis for doing that.
And you have the other extreme. You have the older people who have a very slow immune reaction, immunosenescence.
Yeah, and it would be nice to reassure people who have some hesitancy about vaccines that, no, we've really optimized these things. We know.
I mean, if you tell that to people, we are already pretty in the dark about those adjuvants. I mean, you understand the hesitancy.
Yeah, well, there's a lot of experience with them.
Right.
So, thank you to the people over the last 100 years who've had aluminum salts jabbed into them that we know that it's pretty safe.
So you found that those adjuvants actually slow down the system, right? So create more inflammation.
Well, they slow down the lymphatic pumping. What we found is that all three classes of adjuvants actually slow down lymphatic contractions. So they slow down the delivery of information. Maybe what the lymph node needs is a gradual delivery of the antigen. And so slowing down the pumping spreads out the delivery of the antigen so that more cells can take it out. When we first did the work, we expected that they should increase lymphatic vessel pumping to get the information into the lymph node as quickly as possible. And that's just not the case. So this really suggests that there's a lot that we don't understand about information delivery into the lymph node.
I have 3 questions here. Number one, if we put the vaccine in your arm, the pumping is locally slowed down or the whole body slows down?
I don't know. The experiment we did was an experimental system on a single isolated vessel, as you saw earlier. I don't think anyone's ever looked at that, but it could be out there in the literature somewhere.
But it's short term, right? You just slow down for 1 or 2 days.
We don't know that either.
And when you said it's slow, do we know how much slower it is than physiological state?
Yeah, I think the flow rate would be about half what it would be in the normal state.
So let's say you double the time of reaching a lymph node, you still reach a lymph node within reasonable time, right, to get a reaction.
Yeah, likely minutes. But I think if there is a benefit to slowing down the flow, it's that instead of giving an initial bolus of antigen that then drops off quickly, you get a more gradual rise and a more sustained delivery of antigen over time.
So that's also in the theme of more exposure, a longer exposure of the antigen to the nodes, and so a stronger immune response.
Yeah. To me, that would make sense. Yeah, so this is where we're headed with some of the lymph node work. So first of all, you can see on the right there, the spinning depiction of blood vessels inside a mouse lymph node. So a mouse lymph node is about 1 millimeter in diameter. So these are color encoded by size. So one of these big red vessels is an artery and the other one is a vein. And you can see they go all the way through. The capillaries are in blue here. So there's a lot of capillaries for fluid exchange, for cellular exchange. And the high endothelial venules would be probably in the light blue or green color in one of the branches. It's a fascinating structure of blood vessels inside the lymph node. Most of the capillaries are concentrated in the periphery of the lymph node, not in the middle. We don't know why that is the case, but it'd be interesting to know. So again, as we've talked about, there's important fluid exchange between the lymph compartment and the blood compartment. That all happens across the walls of these blood vessels that are shown in this movie here. There's cell transport across the walls of those vessels as well, primarily at the high endothelial venules. And of course, all of this changes with vaccination. It changes with cancer. So lymph nodes swell after vaccination. Lymph nodes swell when they get the metastatic cells in them. The blood vessel structure remodels in response to all of that. I wish the science knew more about why this happens. What is the benefit of lymph node swelling? What does it do? I don't think anybody really knows. And so we've done some computational simulations that point to a couple of different possible reasons why lymph nodes swell during a vaccination.
So what was your conclusion? What did you find the lymph node swell?
We think it has to do with the maintenance of T cells within the lymph node. So there's a natural turnover of T cells in the lymph node. So you think about it, there'll be some T cells coming in, there'll be a certain number that are resident, and a certain number of T cells that leave. And there are lots of signals, biological signals for the cells to come in from the blood vessels. There are biological signals that tell them to stay there, and there are biological signals that tell them to leave. And it's the balance of those biological signals that can get changed with a vaccination, let's say. So if there's a trigger for more T cells to come into the lymph node and less of a signal for them to leave, which we think is actually the key part, is that the signal that tells T cells to leave the lymph node gets shut down during vaccination. You need to swell the lymph node in order to make room for all those additional cells. It's harder to retain the T cells in the lymph node unless you swell it.
Well, thank you so much, Jimmy. It was
Thank you. Can I just show quickly? Actually, I got to acknowledge all my collaborators and Ph.D. students that worked on this stuff in our funding sources. We just couldn't get anywhere without the brilliant people that actually do the work, and of course the support. So I just want to add that at the end, if that's okay.
Well, I hope you're going to have a lot of interesting grants, because it's so interesting.